Okay, this is the third video and we're going to be working on tutorial 6, which is the first quantitative genetics tutorial. Okay, so this is a very simple tutorial, and I won't focus too much on the details because we all need to work as quickly as possible, and this is very simple. But I'll try and explain some of the concepts a bit more easily. Okay, so for question 1, let's write that here. For question 1, it asks us two inbred varieties of a plant are crossed and they give variance in leaf number in the F generation as 1.5. Okay, the variance in the F2 generation is 6.0. What are the genotypic and environmental variance components and the broad sense heritability? Okay, so we need to take into account a couple things now. It first says that two inbred varieties of tobacco are crossed. So we know that true breeding requires two inbred or two sets of individuals who are opposite homozygotes. So one homozygous dominant, one homozygous recessive. So this kind of suggests a similar sort of situation, except it's two different lines, not just two different individuals. So if we use the example of a Punnett square as an analogy, this is not describing what's actually happening, just an analogy. We know that if we cross two true breeding individuals, we should get 100% heterozygotes, okay? So, technically, you could say that the entire of the F1 generation has the same genotype. Their genetic content should be exactly the same. Alright, so in the F1 generation, it says, according to this question, there is still variation to the degree of, sorry, let's put the F1 variance, to the degree of 1.5. Okay. If the genetics are supposedly exactly the same, then what is this variation attributable to? Well, considering we know that the total variation, if we call it VT or VP, doesn't matter, they mean the same thing, is made up of an environmental component and a genetic component. And we know in this particular situation that there should be no genetic variation, yet we are still seeing variation in the phenotype. Then we can only say that it has to be attributable to the environment. So therefore, straight off the bat, we can say that our environmental variance is equal to 0.5. Okay. Then they talk about the F2 generation, that they have variance to the degree of 6.0. Now, if we continue with our Punnett square analogy, if we were to cross the F1 generation to get an F2 generation, so if we take these heterozygote individuals and cross them, we should get one part homozygous dominant, two parts heterozygous, and one part homozygous recessive. Now there is genetic variation in this generation because there are individuals with different genotypes. Okay, so we assume that there is a genetic component now. But does that mean that there's not an environmental component? Obviously no, because the environment always has an effect on your phenotypic variation. So in this case, we have an environmental factor and a genetic factor. So we're going to call the variation that we see in this generation the total variation that we see altogether, because it has genetic and environmental components. Okay, if we have our total variance, or variation, however you want to call it, and our environmental variation, we can use that to calculate our genetic variance, or our variation due to genetic components, which is the same as our total minus our environmental, which should be 4.5. Okay, 6.0, 1.5. Okay, now it also asks us to calculate the broad sense heritability. Most of you should know this, but the broad sense heritability with a capital H, broad as in big, is equal to our total genetic variance over our total phenotypic variance. Okay, and we have both there. What is it? It's 4.5 over 6.0, and that should give us 0.75. And that is our broad sense heritability. See, pretty easy. Question 2 is, we discussed it in the last video, talking about, it just asks us to calculate the mean, variance, and standard error of the mean in these two generations, given by the values in the table in the question. So I'm going to do the same as we did last time. I'm not going to do the actual calculations, but I'll give you the formula, because you can work them out on your calculator. So the mean, as we know, is x bar, and it is calculated by the sum of all the values divided by the total number of values. The variance, which I explained in the last video, there's lots of ways to write it, but I like to use this one because it's least confusing and easiest to calculate. It's the sum of all the values, which are, after squaring them, divided by the total number of values minus the mean squared. Nice and easy. Okay, and the standard error of the mean, also easy, but sometimes you can't just click a button on your calculator to work it out. It should be, sorry, let me just double check, but it should be the standard deviation, not the variance, divided by the square root of the total number of individuals. So just for clarity's sake, I'm going to put here SD for standard deviation. Okay, so that's all it is to calculate those values for that question. Okay, for question 3, it just asks to list some human traits which could be in controlled by polygenic a polygenic mode of inheritance. I'm not going to go through that because that's in your notes. So, question 4. It says the following variances were calculated for 2 traits in a herd of pigs, and it gives you the phenotypic variance, genetic variance, and additive variance for back fat and body length. So for those 3 variance components. The first part of the question asks us to calculate the broad sense heritability and narrow sense heritability for each trait in this herd. Okay, so we know that broad sense heritability with a capital H is the genetic component over the total variance, and narrow sense heritability, which is small h, is the additive variance over the total variance. Okay, that's pretty easy. You just have to do it for those 2 traits given the values in the table. Now it says for the second part of the question, which of the 2 traits will respond best to selection by a breeder? And you have to give a reason in your answer. Now, before we go on to explaining the answer for this question, I need to explain to you the theory behind this. So, we know that the total genetic variation is made up of the environmental component, and the genetic component. But there are lots and lots and lots of different things which make up or contribute genetically to variation that you see in an individual. And the three main ones generally that we see, so if we're going to write this as an equation, are your additive variance, your dominance effect, and your epistatic components. Now, your additive variance is the variation that you see. You can Google this, you can find a nice definition for this, it's also in your notes. It is the variation that we see in an individual's phenotype attributable to the exact sequence that they have, so their actual DNA. So for instance, you might see a difference between me and my brother in terms of our eye color because I have a different allele to him, for example, and that variation is attributable to that particular component, one that is heritable, one that is actually to do with the genetic material which can be passed on, not just the other parts of protein synthesis and gene expression and all of those things, which are not always controlled by genes or the environment. Sometimes it's a combination, but that's more complicated for now. So this value, this VA value, this additive variance, when put in the narrow sense heritability equation, tells us that this is the proportion of variation out of the total that is directly attributable to what DNA sequence you have. Therefore, if we have a higher narrow sense heritability value, as in you're going to see more variation due to that particular part of your DNA, not all the other effects that could be going on, like environmental and whatnot. That part of that variation is what's going to mostly determine what your phenotype will look like in the next generation. And so therefore, if you have a high narrow sense heritability, in context to this question, the trait which has the higher narrow sense heritability will respond better to selection. And I can prove that by showing you this equation, which you all should know. The higher the h squared value, the higher your response to selection will be. Okay, so there's your proof. Right, for question 5, this is, in fact, I don't think I need to do this at all because it's basically just asking the same thing, calculate the narrow sense heritability and which one will respond better to selection. Okay, so it's exactly the same thing. I won't do question 5. Question 6, however, let's have a look. Question 6 says that in one population, a mean of 8.5 was acquired, and a subgroup of this parental population had a mean of 0.6. So if we imagine this to be our population, those are all of our individuals, and they have a total average or mean of, what did they say, 8.5. Okay. In this case, these values, the smaller the better, because it was the time to be trained to do something. Okay, then what they did is they picked out a subgroup of these individuals with the quickest time. So let's just pretend those individuals had the quickest time. Then the average of that subgroup, let's just call them that or whatever, the average of that subgroup was 6. Okay, so now these individuals of that subgroup interbred and created the next generation. that generation, so the F1, had an average of 7.5. And it asks us to calculate the narrow sense heritability for this trait. Now this formula, most of you should know, as written in your notes, is the outcome of the progeny, or the child, thus TC, is equal to the mean of the original population plus the narrow sense heritability times by the parental mean minus the mean of the population. So if we look at what values we have here, this was the mean of the population, this was the mean of the parents, so this can be our TP value, and this was the mean of the offspring in the next generation, which should be TC. And then all you have to do is solve for h squared, and you should get 0.4 as your answer. Okay, so that's question 6, which is relatively easy, and you'll use that calculation a lot, usually especially in the following tutorials. Question 7. Now question 7 confused quite a lot of people because they assumed that it was similar to the first question of this tutorial, but it added a couple more components. So it says here that the mean and variance of corolla length in 2 highly inbred strains of a plant and their progeny are shown in the following table. One parent has short corolla and the other parent has long corolla. Calculate the broad sense heritability of the corolla in this plant. Okay, so now our traits, they're just explaining that that is our corolla. And it gives us the variance values for a number of things. It gives us the variance values for our first parent, our second parent, the F1 generation, and the F2 generation. Now, if we go back to our first question, it tells us here, according to what we calculated, that our total genetic variation came from our second inbred generation, our F2 generation, because it accounted for any environmental factors that we had seen previously, as well as genetic factors, because the individuals were now had now recombined, the alleles had recombined such that we get different genotypes. Okay, so first things first, okay, let's just write down what these variance values are so that you can see. So this one is 3.12, 3.87, 4.74, and 47.7. Okay, so automatically, first things first, we can call this our total variance. Now, again, looking back at what we worked out in the first part of in the first question, our genetic variant, our environmental variation is what we saw in the F1 generation. Now, that is not quite the case with this question, which is why people got slightly confused when they did this in the tutorial. What you need to understand is that in the first question, they assumed that in the 2 breeding lines of the 2 parents, there was absolutely no variation. The variance was equal to 0. Okay, and that what we saw afterwards in the F1 generation was absolutely purely environmental variation. But that is not the case here, because it already tells us that in our parental lines there was variation before. Now, as we do with these things, we need to consider the parents as one unit, because they both contribute equally to the next generation. So the first thing we're going to do is give a standard parental value of standard variance of parental value, which will be the average of the two parents. Okay. Divided by 2. And we should get 3.5. Okay. Now we can consider that initial parental variation in the parental line before we've even done the breeding to get our F1 generation. Now we do the parental cross and we get our F1 generation, and we see that we have variation to this extent. Now that variation, we have to account for what we see in as a result of the parental variation. And what we'll see then is that the variation that we see in that F1 generation is going to be attributable both to the variation that we saw before as well as the environment. So what we do to calculate the environmental variation from that is to take our parental variation, which we already calculated, and the variation we see in our F1 generation, and we average that as well. And that value is going to be our environmental variation. Now that we have our VT and our VE, we can do the normal thing. We can calculate our VG, which, if you remember, it's VT minus VE. That should give you 58. And your broad sense heritability, which is your VG over VT, you should get 0.91. And that's all there is to it.